Good evening, class. Welcome back to Metric Hub. Today we are going to talk about meiosis. I just want you to just be very attentive and listen and follow all these notes that I'm having right now, right here, to the end. This is one of the most important chapter which every grade 12 learner must understand in order for you to understand and to be able to work out genetics. So we basically have two types of cell division, meiosis and mitosis. Of most important is meiosis because we need to understand that life on Earth is basically facilitated through reproduction. It is meiosis that is responsible for that process. So now I want you to just check here now that I'm just going to introduce the chapter of meiosis. It's going to be very explicit. Diagrams are going to be attached to explain these concepts that we are going to look at. So we do have what is called what is called normal meiosis and abnormal meiosis, which actually sometimes is called non-separation of chromosomes. So now we are going to be looking at all these. So basic terminology in meiosis is actually attached in this presentation. I want you just to also quite a lot follow and be able to understand them very well. These diagrams that you are seeing here, they are key towards understanding genetics. And as much as you know, you cannot be able to go and be balance those probability chance factors and all other things that we are going to look at and be able to understand reproduction. Every other chapter is built upon meiosis. So you are seeing these diagrams here. They are key in terms of those terminologies that we are going to talk about. Just follow. We are going to talk about also the replicated and unreplicated uh, chromosomes, which at the end of the day, we must also be able to understand the basic structure of chromosomes. What do we mean by homologous chromosomes? What is a chromatide? What is centromere? And also understanding the importance of all these. So I'm just going to introduce all this right now without wasting any time. Let's just go straight into the subject of today. Meiosis is basically a very important and special type of cell division that halves the number of chromosomes. In other terms, if you can check in other books, they say that meiosis is a reduction division process. It is defined as a reduction. It is defined as a reduction division process, which I'm just going to let you see right now. So, what happens is that four genetically different haploid daughter cells are formed from one diploid cell. So, what we need to understand here is that it results in four genetically different haploid daughter cells. Daughter cells are the cells that are formed after the process of cell division or after meiosis has occurred. And again, I want you to understand that the number of cells that are formed in meiosis, there are four. You should be able to understand again that it is different from mitosis. Mitosis and meiosis results in different number of daughter cells. Please take note and put a question mark on that. So now, meiosis basically it halves the number of chromosomes. It halves the number of chromosomes. What are we saying? Meiosis, why meiosis is important? Haploid gametes are produced. This is the haploid that we are talking about to say that it results in what? In half the number of chromosomes. So now, what meiosis does is that in a cell, a normal human cell has got 46 chromosomes. It has got 46 chromosomes. So when meiosis occurs, if meiosis has taken place, it divides by two. That is halving. So when we divide by two, we get what? We get 23. When we get 23, this is equals to haploid. that is equals to N. So now what you need to understand is that meiosis it divides by 2 the number of chromosomes. Why is that process important, or why does it divide by 2? It is dividing by 2 in order to form uh, sex cells that are supposed to be used in reproduction. So in this case, the 46 again, I want you to know that that is 2n, which is 2n divided by 2. We have got what? We have got n here. This is the same as to say that you can see meiosis divides by 2, which is 2n as diploid, and then divided by 2. If we divide 2n by 2, that is halving process that we are talking about. It will give us what? It will give us n. The n that we said is the same as what? As 23. 23 and n is one and the same thing. To say is one set of chromosomes. Is one set of chromosomes that is there. So one thing that I just want to emphasize, so that when we are going to do genetic crosses, is this concept of halving the number of gametes, or halving, we are saying halving the number of chromosomes that are within a cell. To say, if we have got, for example, I can as well give you this, for example, to say we have got MM as the total number of chromosomes. So now what happens is that if it divides, if it halves this MM, it is going to give us what? It's going to give us M. I'm sure that we can see that the total number of chromosomes in the cell was MM, and then that was before meiosis, and then after meiosis, we got to what? We have to understand that we have got half, which is M. If we have got XY again, I want you to see another thing. A set of chromosomes have got XY chromosomes. It has got XY chromosomes. So now, if meiosis has to occur again, this XY is before meiosis. When meiosis has taken place, we are going to have the separation of this, which is going to give us X. It is going to give us what? Y. That must be known by a grade 12 student. This is very key, that we are saying that it divides by 2 the number of chromosomes. It halves the number of chromosomes. The XY that we are giving as an example in this case is again identifying the 46 that we have given in that case, which again, for you to keep on, to just give the last example in this case, we have got 8 chromosomes before meiosis. This is before, we are saying 8 before meiosis, and then after meiosis is going to give us what? Is going to give us 4. We are saying before meiosis we have got 8, and then after meiosis it must give us what? It gives us 4. We have got 32 before meiosis. If we have got after meiosis, we must divide 32 by 2. We get what? We get 16. These are key fundamentals, or key concepts and information packs that a grade 12 learner must be able to understand. The meiosis is very important because it produces haploid gametes. Haploid gametes are produced by meiosis. So if you are going to see in the process of reproduction, or in the genetic crosses, or in the genetic flow diagrams, which we are going to have, of course, to work on in the next chapters to come, you shall see that after meiosis, we must be able to identify and see what are we saying, or what have you said? How did you get that? That is one key important factor. And then meiosis, we are saying it is important. The doubling effect of fertilization on chromosome number of future generation is overcome by meiosis. We have to understand that is overcome because of meiosis. Meiosis helps to overcome doubling effect. It overrides doubling effect. What do we mean here? We also say that a chromosome or an organism has got a diploid number of cells, has got a diploid number of cells, which is the full set. The full set is what? we are saying is diploid, which is 8. And then when a gamete is going to be formed, it is the same concept that we say that we are not going to see 8 from the father. We don't have to find a situation where we have got 8 from the father and then we say plus 8 from the mother. This automatically we say that if it occurs, you know that this is a doubling effect that we are going to see. To say the total number of chromosomes that are in an organism is going to give us what? Is going to give us 16. But 16 is an abnormal organism. If we can see here, it will be an abnormal organism because an organism that we are talking about is a normal living organism, whether it is a plant or it is an animal, it must have, we identify and note that it must have what? It must have eight chromosomes. So in the process of reproduction, there mustn't be a case of doubling effect in terms of fertilization. Fertilization does not occur whereby we have got doubling effect in terms of our future generations. So this whole process that we are talking about here, this one that is given here, it is what? It is overcome by meiosis. It is overcome by meiosis. How is it being overcome? It is X'd by meiosis because meiosis divides by 2 in order to form gametes. Gametes have the number of gametes are going to be what? They are going to be haploid instead of them remaining what? Instead of them remaining diploid. So meiosis results or allows uh, haploid cells to be formed that are used in what? In reproduction and fertilization in the same sense. That is what we are referring to as avoiding doubling effect. And then it introduces genetic variation in living organisms. Living organisms must be very different. I'm sure that in life science students, we have heard of the term or we have heard of survival of the fittest, that organisms have got different strengths, has got different characteristics. They do have quite a lot of variations that exist amongst them, like differences that exist amongst them. So meiosis introduces genetic variation. How it introduces that through what? Through it allows that through crossing over. Crossing over occurs because of meiosis. It also allows random assortment, random arrangement of chromosomes is also facilitated because of meiosis. These two processes are key towards what? They are very important in regards to genetic variation. So when organisms have got different characteristics, have got different strengths, have got different appearances and whatsoever, we say that it is because of the variation of genes that are in them. So that allows or that promotes survival of organisms. Organisms are gonna be able to survive because they can be able to bypass uh, limiting factors that might be exposed upon them differently. Instead of them to be wiped off at the same time, they cannot die at the same time, but they are going to endure and survive different environments at different capabilities. I want to look at key terminology that we need to know when we get into meiosis, chromosome. Sometimes people would come and see quite a lot of times we talk about quite a lot of times we talk about this chromosome. What is a chromosome? What is a chromosome? So chromosomes, I want you to note that they are thread-like structures made of DNA and protein found in the nucleus of living organisms, what they do is that they carry genetic information. They carry genetic information in the form of genes. We say that a person or organisms, when they reproduce, characteristics are able to be passed from parents to offsprings because of chromosomes. Chromosomes, they are protein-like structures that actually carry the DNA. The DNA actually is the one, the deoxyribonucleic acid is the one that carries the genetic information, which we say these genes are found in DNA, or we are saying chromosomes enables succession of characteristics in a population species. That must be taken note of. Let's just take note of that. We need not to I don't want anyone to miss anything right now. We need to be quite aware of that as we continue. So now, chromatide, one of the two identical strands of a replicated chromosome. We are saying when we look at a chromatide, we consider that as a single piece of a chromosome, which is identical strand of a replicated chromosome. So I'm just going to show you this or such is explained or is shown in the on the board here through some diagrams. And then centromere, if you can check here, the chromosome, we are saying they are long protein-like structures. As you can see here, if we take two pieces, as we can say that it's like this, and that when such a structure is formed, this is the same as the one that you are seeing here. It is identifying two chromatides that are joined together. When two chromatides are joined together, they form a chromosome. So a chromosome you are seeing, like in this case, it's a chromosome that is shown these two black pieces that are two pieces of chromatides joined together. They form a what? They form a chromosome. So now a chromatide, as you can see, is a single piece. When we say a chromatide, it only identifies the single piece. The single piece of a chromosome is what we call a chromatide. One of these two pieces untied, we say that is called a what? A chromatide. As you can see here, the chromatide is given there. So in exam, sometimes you shall find out that the question would come asking you to label. It will be asking you to label the following: X, Y, Z. X is the chromosome, Y is the chromatide, Z is the centromere. So the centromere is actually region where two chromatides of a chromosome are held together. The white piece that is tying, as you can see, this number 3, we are saying it is the chromatide. I mean, it's the centromere. The centromere is a region, or it's a piece that joins together two chromosomes or two chromatides together in order to form a chromosome. So that is a centromere. So as you can see here, we are saying if these two chromosomes lie next to each other, we say this is called a homologous chromosome, one from the mother and one from the father. When they lie like that, we say these are called homologous chromosomes. That is number 4. So if you can look at this, you should be able to understand and be able to label these and also define and explain what each of them is. Homologous means of the same type. Homo means same. So these are of the same type, or two chromosomes that are one from the father and one from the mother, containing similar genetic information. However, what happens there is that, as you can see, one is black, one is father, is white. So we can say paternal, and then we can say this is maternal, as to say from the mother. So as you can see there, we say as they lie together, if we have got a paternal and maternal chromosomes next to each other, we define them as homologous chromosomes. They are called, or they are defined as homologous chromosomes. I want you to always note that. these terms are always going to be used continuously in this unit. And then bivalent, bivalent, a pair of homologous chromosomes which lie next to each other and physically are in contact with each other at a point where crossing over will occur. Crossing over, I'm just going to demonstrate this also in the next pictures that we are going to see here. But the question and point is that when a pair of homologous chromosomes lie next to each other and are physically in contact, what we need to know is that a process of exchange of genetic information defined as crossing over is possibly going to take place. It's going to take place. So that is a bivalent. That is what we call bivalent. And then unreplicated chromosome. NB, this unreplicated chromosome, I want you to observe quite openly here. Unreplicated chromosome has a single double-stranded DNA molecule. If I was in class, I was going to ask someone to just demonstrate what we are talking about. But for the sake of progress, and I'm here to show you that just be open-minded, because you shall find questions which are going to let you or to ask you, how do we say that or differentiate between the two cells? How do you say that this cell has gone through DNA replication or DNA replication has not taken place? How do you distinguish a cell that has undergone replication and that has not? So in order for you to know that difference, you need to know unreplicated chromosome. How does it look like? Unreplicated has got a single double-stranded molecule of DNA. So what happens there is that it is more like to say that it has got a single strand of a what? Of a chromosome, which we say that sometimes we can say that it has got a chromatide. It has got a chromatide somehow, somewhere to say. So now, what we need to know is that this is a single double-stranded DNA molecule, which is, we call unreplicated. It is called unreplicated chromosome. There's a diagram here that you are going to see. And then we say replicated has two identical double-stranded DNA molecule. It has got two. And then unreplicated has got what? Has got one. If it is what? Unreplicated. I'm sure that you remember replication. I just had a lecture last time of DNA replication, producing a copy identical to the original. We call that replication. Multiplying multiplying a single. We are saying that process of replication has to have or produce an identical cell that is of the parent cell. So now, see here, unreplicated chromosome. This is shown as what? As unreplicated. This is shown as unreplicated class. So if we can see a chromosome, a chromatide that lies alone or that is separated, we call that is what? It's an unreplicated chromosome. And then DNA replication results in what? In replicated chromosome, which results in two chromatides being joined together. So we see chromatides joined together. When they are joined together, therefore, we say that it's what? It's replicated. It defines or gives a replicated what? Chromosome. It gives a replicated or results in a replicated chromosome. That I'm sure you are seeing. Identify diagram A versus diagram B. You can see what I'm talking about. Diagram A has got a single, it has got a single strand or a single piece that is untied. But on diagram B, you see that we have got those two. I have got these two pieces that are tied together, which I can say piece one, and we have got also piece two that you can see there. So unlike in diagram A, we only have one piece here, which is alone. But piece two, it ties together two pieces of one, which results in B being what? A replicated chromosome. It results in a replicated chromosome that you are seeing there. And then we also have the terms like interphase, the phase in the cell cycle when DNA replication or where DNA replication occurs. DNA replication occurs during interphase in meiosis 1. So now it is a cell stage. It is a stage in the cell cycle which allows the replication of DNA. You shall note that this must be taken note of again to say when do we have DNA replication, or at how do we, which stage of meiosis does DNA replication takes place? It takes place during interphase, and then what happens is that it is a stage in the cell cycle where there is multiplication of DNA. The multiplication has to increase the DNA contents to prepare for cell division. That is one thing, again, I want you to take note of. We have got the stem diploid, which is also given as 2n. I also told you to say, if we say 2n is equally the same as what? As diploid, is equally the same as what? As 46. I will normally want to use the number of chromosomes in human beings. That is simple because the one that is quite known, if I talk about any other organisms, you might be confused. Let's use 46 as the most known one, number of chromosomes. So diploid is 2n, which means two complete set of chromosomes. Where is the two complete set of the chromosomes? We are saying this is coming from where it is coming. It is coming from n of the father. We said n plus n is equals to 2n. The other n is coming from the first n. It is from the mother side, and then we have got the other n, which is coming from the father side. So now we have got the genes of the father, we have got the genes of the mother as well. So what are we saying? This is fertilization. Diploid cells are formed after fertilization. It means fertilization has occurred. These are resultant cells of what? Of fertilization. They are resultant cells that are formed they are resultant cells of what? Of fertilization. Please note as well, that is one thing that you need to take note of. And then the opposite of that is what? Is N. N, one set of chromosome in the cell. In terms of N, please note that the N is it refers to only one side. It can be N is equals to either, we are saying it is either the mother, or the father. And therefore, what it means is that this is formed because of meiosis. The haploid cell is a resultant cell formed after meiosis has occurred. Is formed after meiosis. Is formed after meiosis. Is the one that we are talking about, as to say that it is 23, as I said there, to say that it's a what? It's a sperm cell. it's a sperm cell or it's an egg cell that has been formed in order to prepare for fertilization, in order to allow reproduction. So a haploid cell is either a sperm cell or it's an egg cell which only has half the number of chromosomes of the organism. In that sense, we say that it's a normal cell of an organism that can allow fertilization. Then we have got gene is a segment of DNA in a chromosome that contains the code for a particular characteristic. Characteristics are carried by genes. This is quite common expression that we have seen in our societies to say you look alike to your mother, you look alike to your grandmother, and then people have to explain it because of the contents or the genes that you have to say that the person, the child looks alike to so and so. The alikeness is because of the genes that has been inherited by the person who looks alike to that other one. So the genes are the ones which are a segment. We are saying it's a segment of DNA that contains the code for a particular characteristic. Characteristics are carried by genes in living organisms, which again, these genes are a portion of DNA. They are a portion of the DNA that we are going to continue talking about as we go there. And then we have got central structures found or formed when the centrosome divides into two. They move to opposite ends of the cell during cell division. I want you to look at the diagram again. I have came with diagrams to help to explain that, to say that when we talk about the centriole, you can see what happens is that there are structures which move to opposite ends of the cell during cell division. So you see that that is a what? That is the centriole. So I'm just wanting you to see that quite a lot of times we come to exam or we met in exam questions such as this is just a structure of a cell which you must be able to label and give functions of them to say what is this, what is what. So the spindle fibers, these are thread-like structures where chromosomes or chromatids are attached. This actually controls the activities of the cell in order for different stages of meiosis to take place. They are controlled and regulated by the expansion and contraction of the spindle fibers, influencing the cell functioning, which at the end of the day facilitate the process of meiosis. The spindle fibers that you are seeing here, thread-like structures that are attaching from the centromere to the chromosomes and also to the other opposite central. So now what you need to take note of is you need to know what is spindle fibers, what is centriole, and then this is the cell membrane. It must be taken note again. We have to talk about, of course, we say the chromosome is as a whole. This chromosome is looking at this as a whole, not only one piece. The chromosome is looking at this whole structure as a full set. It's a chromosome. I told you that one piece, this one alone, it's a chromatide. We have looked at it and labeled it just now. I'm sure you know that is a chromatide. A chromosome is when it is all of it. And then we can also be able to look in the process, the stages of meiosis. Let me not discuss about the stages as to say, otherwise, what does which stage is being represented by this diagram? So, as I have explained there, I was trying to also attach diagrams which would enable students to visualize, and most of the times it's better for you to see than to be told. I want you to see, just see and not. So this is showing a diploid 2N. This is a diploid cell. When we have got this together, we say that these are diploid. These are diploid. It's a diploid cell. And then haploid, you can see it's separate. It's separate. So if we look at this alone, this one, it identifies that the cell is what? This alone, this one. If we look at this one as it is in that structure, it shows that it's a it's a diploid cell. I wanted to be seeing like that. And then obviously the other one also, which we can give to say it is uh covering this piece. It is looking at this white one alone. White one alone is also a 2N. We are saying the black one also is a 2N. If you look here, we are saying this one is an N, and then the other one again is an N. So what is happening is this is haploid. This is a haploid, and then obviously the opposite is also correct to say that that is diploid as indicated by what? By the board, on the board there. And then you can see again the relationship between the gene and DNA. So you see that this is a chromosome, and then chromosomes are tied on a what? Chromosomes are tied on, we are saying that this piece is just extended and shown to say that this is the same piece that you are seeing. Chromosomes are they carry the what? They carry the genes. So genes tie themselves on chromosomes. So as you can see, the DNA structure, it is the one that is carried by the gene, which is explaining the point that I was looking at, this one of saying what is a gene and what is a chromosome. Chromosome, long thread-like structures which carries the genes, and then you can see on the diagram there to say that is exactly what I'm explaining, what I'm trying to show you here. And then crossing over is the overlapping of homologous chromosomes resulting in the exchanging of genetic material during prophase 1. When does crossing over take place? It occurs during prophase 1. What happens? Homologous chromosomes, they lie next to each other, and then they exchange the genetic information. They exchange their genes, they exchange their DNA. So it occurs during prophase 1. And again, I've also made it away. I've also put this other next term again related to it to say what is chiasma. It's a point where two chromatides overlap during crossing over. The point of contact where two chromatides are exchanging the material. We say that is called what? The chiasmata. The point of crossing over is a point of contact. Point of contact between homologous uh, point, it's a point of contact. Why does that contact has to take place? The contact is occurring for allowing or to promote what? To allow the process of crossing over. That is one thing that I want you to take note of. As you look at this, I have also drawn a diagram that can also illustrate what I am explaining here. We say these are called bivalent chromosomes. So we say that the point of crossing over is shown by the dotted line that is cycling that point. And then the point of contact, which is shown as the chiasmata, is this point of contact which is taking place. That point of contact, we call that the chiasma. The chiasma is the point of contact between homologous chromosomes, facilitating exchange of genetic information. So as you can see, the resultant chromosomes that are formed, we are saying this is during crossing over. As you can see, during crossing over. Crossing over is being demonstrated here on the diagram, on diagram 1, and then diagram 2 is after crossing over. Is after crossing over or a crossover. So now, how do we say that it is after? You see these two uh, these two chromosomes, the first one was completely white without black, but we see there is a portion of black that you can see here. This one was completely black, but we have also a portion of what? We also have a portion of, a portion of whiteness is also seen on that other piece. A portion of white is seen there. A portion of black is also seen on a complete white chromosome. It was fully white, but we have got a segment of black there. We have got a segment of white here, which is identifying and explaining that crossing over has taken place. And then we say the point of crossing over is the chiasma, which is shown by this dotted line, which is showing this to say it's a point of crossing over. That point of crossing over, it is called the chiasma. I'm sure you can be able to relate this very well. You can be able to see and make sense of this. And one thing that you should always notice that when it comes to exams, these are the fundamentals. These are the major points which examiners would always want you to take note of. And as much as you may have made sense of them right now, as we go, I promise you that everything will be just easy. So we are coming back to this one. Is showing us the karyotype. Is the karyotype that is shown in this at this point is the karyotype, the structure, arrangement, and appearance of chromosomes of an organism. We call that what karyotype. I'm just going to define it and give you here. It is the representation of the number. We have the number, we have the shape, and also the arrangement of a full set of chromosomes in the nucleus of a somatic cell. That is called karyotype. When we look at the arrangement, the representation, the shape, and the number of a full set of chromosomes in the nucleus of a somatic cell, we define that as the karyotype. As you can see, this is the karyotype that we are looking at. It is the number, how many are they? The number, in terms of the number, is equals to 23. We have got 23 pairs. We have got 23 pairs. That is the number there. This is the number that we have got 23 pairs there. And then the arrangement, you can see, we're seeing from number 1 to number 23 there. And then we're saying the number, the structure, the shape. In terms of the shape, again, I want you to note that the shape, if you can look on the shapes of these chromosomes, they are not equally identical. I mean, they are not identical to each other. We have got this number 23, it's got its own different shape, which is different also from number 4 and also different from number 17 and so forth. So these chromosomes, or we see this diagram, it represents the karyotype. It represents the karyotype, which I have defined as you have heard now. Still on the same diagram, we are also supposed to note what is called autosomes. Autosomes, the first 22 pairs of chromosomes which control the appearance, structure, and functioning of the body. These are called autosomes, the first 22 pairs of chromosomes on the karyotype. They control the appearance, the structure and functioning of an organism. These first 22 of them. So if you can look on this diagram, we are saying from number 1 to number 22 is equals to what? Is equals to autosomes. These are autosomes. And then one thing again that you must understand is autosomes. What is the function of them? What are the functions of autosomes? So if you can see here, you see that the autosomes, they are there to control appearance, the structure and functioning of an organism. So they are controlling every other bodily functions or every other bodily features, which we can see that they control the other bodily features other than one thing that is left. So we are saying from this one up to this one, they are all called what? Autosomes. They are called autosomes. And the functions, we say the structure, the appearance and functioning of an organism. Structure, appearance, and functioning of an organism is controlled by what? By autosomes. These are chromosomes from number 1 to 22. And then we have got gonosomes, which are called sex chromosomes again, the pair of chromosomes X or Y responsible for sex determination. As we go back again, I'm saying gonosomes define the sex determination. I want us to also check back again. If you can see here on this 23, we say this is defining the 23 identifies, 23 identifies, it identifies the what? The sex chromosomes. It identifies the sex chromosomes, the number 23. And then again from there, I want you to note that when we look at gonosomes, when we look at these gonosomes, when we look at these sex chromosomes, what happens is that you shall find that X chromosome is longer. It is a longer chromosome than what? Than Y. So this is called an X. It's an X chromosome. And then the shorter one, X is what? Is long X chromosome and short Y chromosome. That is important for you to note. So now, if we see a structure like that on our karyotype, this identifies that a man or a male person is going to be, this is going to be male. And if we have got all of them are long, this is going to be what? This is going to be female. I'm sure I've already given you more detail here now that you should know. Questions would come where you are asked to say, what is the gender of the karyotype in question? Identify the gender. Identify the gender in question on the karyotype. So we are saying the gender, if it is like that, it's a male. And then if it is all long, it is all long is equals to what? It's equals to a female. Are we together there? I'm sure that we can see. So if you can look on the diagram of the karyotype that I have attached here, this is going to tell us that this person is what? Is a male or is a female. So in this case, our karyotype that we have given here is for a male somebody. This is for a male somebody because we have got a short Y chromosome that is given on chromosome 23. Are we together in this case? I hope that you are going to understand and make sense of it. Still on the karyotype, it is going to take us again into other questions, which is called a Down syndrome or non-separated non-separated chromosomes that have resulted in a Down syndrome individual. So we shall find out that the person can be defined to say, is he a normal or abnormal based on the karyotype, where you are going to see that on chromosome 21, chromosome 21, 21 chromosome might give you, it might give you, instead of 2, it's going to give you 3 chromosomes. If they are going to be 3, we say trisomy 21. That is called trisomy 21, and then it is resulting in what? In Down syndrome. And then what caused that? It has been caused by nani? non-separation of chromosome 21 during meiosis, non-separation or non-disjunction. This is again another subject of note to be understood when we come to karyotype. Trisomy 21, three set of chromosomes 21 identifies an individual who is, in this case, we should note that the person, this is equals to an abnormal or a Down syndrome child. We are saying it results in a Down syndrome. It's a Down syndrome child who is having three sets of what? Of chromosome. You see 1, 2, 3, which is called trisomy 21. It's a Down syndrome resulted from non-separation of chromosomes that I've said just right now. That is one thing, again, which comes when we talk about what? When we define karyotype. When we define karyotype, you should be able to explain and relate to all that. And again, we now want to look at somatic cells. Somatic cells, these are body cells also. Somatic cells are also called body cells, which is any cells in an organism excluding the male and the female, gametes. They are diploid, two sets of chromosomes, and are produced through what? Through mitosis. Mitosis, we say it is what? Cell. It is resulting in cell multiplication. It is resulting in cell multiplication. So somatic cells, it is resulting in cell replacement. Somatic cells are bodily cells. We are looking at these as any other cells other than the sex cells, which is either male or female gametes. Those are called somatic cells or body cells. And then we have got sex cells. Sex cells, obviously, this defines the specialized cells called gametes, which I am sure that you know that we have got the sperm cell and the egg cell. They have got a haploid number of chromosomes. That is to say that they are produced through what? Through meiosis. If you can look, this is very deep and classic understanding, which should be reflected by a grade 12 student to say somatic cells are what? They are diploid, meaning to say that they are having a full set of chromosomes. They are having a full set of chromosomes. They are produced by mitosis. And then the gametes are specialized cells that are produced by meiosis, which again, the moment you identify that meiosis is equals to N, if it produces anything, it is equals to what? Is equals to half, which is equals to 23. Class, I'm sure that you must be able to understand this, and if you get this right, everything again coming in the next lessons, it will be a walk in the path. So please, let's pick that and let's note it very clearly. And then lastly, I want us to look at non-disjunction, or we say that this is called non-separation. This is called non-disjunction, or can be called non-separation of chromosomes. Non-separation of chromosomes. When homologous chromosome pairs fail to separate in meiosis, when they don't separate, we say that it is called non-disjunction or it's called non-separation. As you can see, we are saying there are homologous pairs which must separate here. So no more meiosis has taken place, which we are going to find this is a diploid. This is a diploid, and then again, that is a diploid cell that is formed there. So now, what happens is that we need to have, we need to talk about the formation of gametes, which we are saying that the cell must divide in order for it to give us what? It must divide so that we can talk about a cell which have taken place. So if you look in this stage, we say that we have got normal meiosis that has taken place. However, when we come to this stage here, we are saying this cell what happened is that it is not it we said last time that if a cell is like this, it's called what? This is also this is diploid. This is a diploid. It's a diploid chromosome. This is a diploid chromosome. So what happens is that it must separate by the chromatides, which has been shown here to say we see chromatide 1 and we have got chromatide 2, which at the end of the day, they've separated into two cells. We have got one cell and two cell number 2, cell 2, which we are seeing that it is containing it is showing an unreplicated, or it is showing a haploid cell which has got only one piece. But if you can see this one, it has got the full set. So now what happens is that in cases when, in instances whereby this cell, can we name it A? And then cell A, if in some cases it happens that it fertilizes or it is fertilized by cell B, when we see that fertilization has occurred, fertilization taken place between cell A and cell B that you see there. What are we going to have? We are seeing cell A, it is going to say cell A that you see there, cell A is given as 2N, and then it is combining with cell B, which is what? Which is N. So what happens is that when fertilization occurs in this respect, we are going to see that we are adding the numbers here, class. I need you to take note that this is just a cell. But what is important is we are saying the contents of cell A is combined with the contents of cell B. So what is being added is equals to 2N plus N. When we add 2N plus N, I'm sure you are mathematicians, and this cannot give you any stress. 2N plus N is going to give us, it is going to give us what? 3N. 3N is what you call trisom condition, which we say that the trisom condition is what you call, it results in what? In Down syndrome, which we are saying the Down syndrome is a result of what? Non-separation. Which cell did not go under, which cell has have undergone or have experienced non-separation. Non-separation has taken place on cell A. Cell A is the one that did not separate, and then it came with its full set of diploid structure or diploid contents. When it came with the diploid content like that, what happened is that it came with its all full, to say that to N, it has been added to N. Therefore, we ended up with a what? With a 3 set of chromosomes that are found in set in the what? In this zygote. This zygote which is formed here, 3N, the zygote formed here is a what? It's having 3 sets of chromosome. It is having 3 sets of chromosome 21. It is having 3 sets of chromosome 21. That is what I want you to actually observe and note. So I hope that you have understood this and it has made sense to you. I wish you all the best. Please keep following the channel. I'm sure that if we can see, we are going far. And I want to put you at the best footing or at the best position, which can make you to build the better understanding of life sciences. Life sciences is quite an interesting subject. Let's do it together and keep following. Please join membership because revision is going to be done extensively. Many other supporting formal and informal work, which helps you to understand what has been taught or what you are supposed to know in a term or in a semester or in a year is going to be covered during relevant times. It is only important for you to follow and make sense of this as of now. So thank you very much. Until we meet again, you turn your notification button so that we can be able to uh, get all this without missing anything. Have a beautiful day. Thank you so much. Keep connected. Share to friends, share to classmates. Let's share to everybody who has to know that Metric Hub is there, grade 8 to grade 12. Give to your brothers and sisters. Let them come and join now. By the time they write Metric, it will be done. Have a good day. Thank you.